Welcome back. Now let's try to look at this very important but at the same time interesting concept, and that is meiosis. And we have to first diagrammatically see the 5 stages of prophase 1 of meiosis. Now let's take a quick revision. Now when we were talking about meiosis, we know this is reductional division. It means when you have 4n, it will be divided into 2 and 2, and that was mitosis, wherein the 2n chromosomes became 4n, they divided as 2n to n. But when you talk about meiosis, well, the 2n chromosomes will divide as n and n. The 2n will become 4n and they'll divide into 4 cells with n, 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 n. That's all haploid. And in this meiosis, we have 2 stages very clearly, meiosis 1 and meiosis 2. Now when you talk about meiosis 1, well, in this meiosis 1, we have prophase 1, which has itself 5 stages, and they are the highly essential parts of meiosis. Why? Because in this prophase 1, we have the concept of genetic overlapping, because of which we have a lot of variations. So why do we have qualities? Why have we inherited things from both mother and the father? Well, that's because both of their genes have overlapped. Both of the chromosomes have overlapped. They have exchanged and they have created something new, thereby creating variations. And this is one of the major star processes in meiosis. So let's try to first draw the diagram for the 5 stages of prophase 1. And in this, firstly, we have the nuclear membrane with the chromosomes which are thread-like, and then we have the centrioles. Now, these thread-like chromosomes, they start to condense and then they start to appear, and the centrioles start to move apart at the poles. In the third process, they are completely apart, and spindle fibers, they appear along with this. We have the non-sister chromatids, that is, the chromosomes, they start to overlap. Do you see that overlapping, the blue and the brown? Well, exactly like that. Then, when they are apart, this complete genetic overlapping has now caused the formation of 2 new chromosomes with new characteristics, and then they'll be pulled apart. Well, these are the 5 phases or the 5 stages of prophase 1. Now let's try to understand this. So prophase 1 is the most important stage of meiosis and differs from the normal prophase of mitosis. It is divided into 5 stages, namely leptotene, zygotene, pachytene, diplotene, and diakinesis. Let's see what leptotene is. First, let's take the word and let's break it down as lepto, L-E-P-T-O-tene, T-E-N-E. Similarly, zygotene can be broken down as zygo, Z-Y-G-O, T-E-N-E, teen. Pachytene will be broken down as pachy, P-A-C-H-Y, T-E-N-E, teen. And we have diplotene, which can be broken down as diplo, D-I-P-L-O, T-E-N-E, teen. Now, when you talk about all these 5, it is also going to involve essentially the crossing over and the recombination of genetic material between non-sister chromatids. That is, the different chromosomes. Now, when you talk about phase 1, that's leptotene, well, in this stage, the replicated chromosomes, the already chromosomes which are replicated and their chromatin network, they start to condense and they start to convert into thin thread-like chromosomes. Now, when they convert into those thin thread-like chromosomes, we have the phase 2, which is the zygotene phase. And in this, the homologous chromosomes, they come closer to each other. And when they come closer to each other, this pairing of the homologous chromosome is called a synapsis. Well, this is very different from the synapse of the neurons. This is synapsis. This synapsis is nothing but when the chromosomes, which are homologous, they start pairing. Then we have the phase 3, which is called as the pachytene. Now in this packaging, the sister chromatids, they become visible, and of course when they become visible, they start crossing over. That is, one chromosome comes over the other where there is a sequence of homology. When they see that certain sequences are similar, they come and they sit there. And this occurs between the non-sister chromatids of the homologous chromosome. Well, the crossing over, the recombination, that point is called as the chiasma, also called as the chiasmata. And this phase of packaging is the one which is going to lead to genetic recombination and of course lead to variations. Let's take the word chiasma and let's try to break it down as chias, C-H-I-A-S, ma, M-A. Now when you talk about the phase 4, which is the diplotin phase, well, this is the stage in which the recombined sister chromatids, that's already that they have exchanged their genetic material, they will start repelling, and the nucleolus and the nuclear membrane, they start to disappear. Now they are starting for another set of the phase. That's phase 5, which is stereokinesis. In this, the chromosomes, they further condense and shorten, they go apart, nucleolus and nuclear membrane completely disappear, spindle fibers, they begin to appear, and when the spindle fibers begin to appear, that is the time they will now undergo another phase, that's metaphase 1 of the meiosis.